Hey, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola, ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 154th seminar, and I call it a Michigan State University Day. I am in New Zealand now. It's my 25th day of this 1 month long trip to Spain, Portugal, and New Zealand. I have been enjoying my trip a lot, giving talks, discussing ideas, and having fun with great people. And actually, yesterday, I also done one thing, I always thinking about, but it took me 50 years to do skydiving. It was fantastic. However, everything has its end, and I'm also look forward to coming back to San Diego next week. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Professor Seung Yon, or in short, Rhee. She is a professor at Michigan State University and also the director of the Plant Resilience Institute. I met her, I think, for the first time last year at the DOE JGI meeting after finding out she and I will be leading NSF Global Centers on two different but complementary topics as the one of the directors. We exchanged many collaboration ideas over dinners, and I look forward to the synergy between the two global centers. Professor Lee, thanks so much for your amazing contributions to the scientific community and also education, and the virtual podium is all yours now. Thank you again.
Great, thank you, Dr. Moon. Hi, everyone. I'm going to share my screen. It's a pleasure to be here. I'm going to try to convince you in the next few minutes the importance of plants and understanding plants in our world today, and that the synthetic biology in plant science is a huge space that's really filled with very few people. So it's a great space for innovation and movements happening in going forward. So this slide is to show you that it's really important to think about the climate that's getting warmer and warmer and the impact of that global warming on food security. So if we have a world that's 2 degrees Celsius warmer, we're going to have close to 2 billion people in addition that will face hunger. And if the temperature is 4 degrees warmer, then we would have close to 2 billion people that will be hungry. And so um, as I mentioned, we all know and feel that the world is indeed getting warmer. In fact, a couple of years ago, 2023 hit the record on being the warmest year, since mid 1800s. So many of you may not know that the food that we take in really depends on really 4 plants. It's wheat, rice, maize, or corn, and soybean. And the number in parentheses represents percent of calories that we take from each of these 4 plants. You can see it's more than 50% of our calorie intake. And these plants, even though they're so critical for our food security, are not very healthy when the temperature goes above 35 degrees Celsius. So in fact, I have here the temperature at which, here, I'm going to just make a laser point here. the temperature at which each of these plants really suffer their growth. For maize, it's 35, wheat 34, rice 33, and soybean is even more sensitive at 32. This is in a review that has just been accepted at Journal of Experimental Botany, and I suspect it will come online very soon. And so temperature is continuing to rise. And as you can see, this is a figure that shows the number of days per year when the temperature goes above 35 degrees Celsius. Okay. And we are around here right now. Okay. That's about 20 days, I would say. But as you can see on this graph, we go further into the future, the number of days that we're going to be experiencing temperatures above 35°C increases. And this is average. Some years there's going to be a lot more, some years maybe less. But if the growing season and especially flowering season hits this or one of these days, we're going to be in trouble in terms of food production. In addition, we lose another 20 to 30% global crop production because of pests and pathogens. This is from a beautiful paper that was published in 2019. Where the authors actually interviewed farmers around the world and identified the major issues in terms of pests and pathogens, what the sources are. And as you can see, for rice, maize, wheat, soybean, and potato, the biggest problem is fungus, shown in red here. And the axis is the global crop loss in percentage. So we lose about 15 to 20% just due to fungal infections. Okay. And for other for the crops, the other sources of loss come from insects or nematodes and so on. Okay. In addition to pests and pathogens, whose disease load is going to increase as a result of increasing temperature. So this is actually going to be even more than 20 to 30% loss in the future. We lose another 30% due to drought. So the image on the left shows you the global drought. This is for, I guess, 3 months in 2023. And it's color coded where the darker the red is, the more severe the drought is. Okay, and you can see lots of the world suffering from drought during this growing season of these crops. And on the right, you see where the crop areas are, where the agricultural fields are. And I hope you can see that there's a lot of overlap between these two. So, food security or insecurity rather is a real issue. And it's really felt a lot more in some country than others. And agriculture, even though in developed countries, it's a pretty small portion of the GDP, culture is still a big portion of the GDP for many countries. On the left, I show you the proportion of GDP from agriculture color-coded. So orange is 25% of the national GDP. If you go darker than the orange, it gets to, much higher, like up to 61%. And it's similar case for the number of people employed in agriculture. Even though in places like in Europe, the number is pretty small percentage of all employees. In other parts of the world, it's a major component of the workforce. Okay, and so agriculture still is a big driver of economy today. And if you go beyond agriculture, actually, if you think about food industry, it's actually 10 times bigger than agriculture. Okay, so this is just showing you the contributions of GDP in the US from various sectors, and you can see food is 10%, and the food industry employs about 22 million people in the US. That's about 10% of employment. And so you can imagine, in many countries, food industry takes a huge percentage of the economy. Okay. So our food source is vulnerable because of increasing temperature globally and increasing disease load, drought, and flooding and stuff like this. So what are the solutions? Of course, we have lots of solutions in managing plants in agricultural systems, which I'm not going to talk about here. But in terms of plant science, and both fundamental and applied, really great solutions exist for from plant breeding and biotechnology. I'm not into this very busy slide. Of course, it's actually described in very detail in this review paper that I mentioned earlier. But this slide is just to show that there are some solutions that can be taken through breeding plants and especially crops and biotechnology such as genome editing. However, there are some issues with these approaches. One is that it takes really long time, similar to developing drugs in pharmaceutical industry. Improving plants and crops is a really long process. You know, years to decades to develop new traits, varieties. It takes like even more to regulate them, get them out into the market. Of course, we have to also contend with negative public perception, especially of things like biotechnology and GMO. And I presume synthetic biology is not going to avoid that either. So we have these are challenges that we have to work on, and there are lots of emerging ways of thinking about and opportunities for addressing these challenges and such as emerging technologies, modeling, connecting more fundamental research with applied research, changing public perception through more engagement, and interdisciplinary and global collaborations. But today I wanted to just point out here that synthetic biology is absolutely one of the biggest emerging frontiers that I think could address a lot of problems, which is the topic of today's main speaker, Adil Khan. So I'll stop right there.
Fantastic. Thank you. This is amazing and very important, as you mentioned. Uh, I'm actually talked to many people here in New Zealand, and unfortunately, you probably imply, a lot of young people leaving New Zealand because New Zealand, main, industry is agriculture, but young people somehow, not that much into it, unfortunately. But it's very important, as you mentioned, and because the we without food basically cannot survive. And you also mentioned the climate change potentially reduce the crop yield, and that's true. And also I experienced uh one of my actually student, actually originally from Africa. He lost two brothers because of hunger and disease in Africa many years ago, and we need to solve this problem as soon as possible. So I'm not sure, I could jump into the plant synthetic biology in near future, but I'm actually doing some potential research that is a plant microbes interaction, including potentially fungi. And I'm excited to know you, and then I'm excited to have potential collaboration between you and me in the future as a center as a whole. So thank you so much for wonderful, amazing talks.
You're welcome. Thank you.
Yep. Okay, so now, main speaker of today with a longer introduction, Dr. Adil Khan is a postdoctoral researcher in the laboratory of Professor Ryan Lister at the University of Western Australia. I thought, Australia is next to New Zealand, but now realize, his time is very inconvenient, midnight, I believe. So thank you so much again for joining us. Adil Khan completed his master in biotechnology at the Australian National University and subsequently completed his PhD in 2021 in plant synthetic biology at the University of Western Australia. And very important topic, as Sue already mentioned. As a graduate student, he worked with Professor Ryan Lister to develop the first reversible CRISPRi-based circuits for programming gene expression in plants. He is continuing as a postdoctoral researcher in the laboratory of Professor Ryan Lister to program uh, plants for space. Importantly, we need more plant synthetic biologists and plant engineers to solve many global problems, as Sue already mentioned, including climate change and food production. I'm so glad to have two researchers today for this SynBYSS seminar working on similar topic, of course, of plants engineering. Adil, thanks so much for your time today. Again, especially very inconvenient time and midnight. Uh, and please take it away and all yours now. Thank you.
Thank you very much for giving me the opportunity to present this work, and I will share my slides now. I should stop and then. Yeah.
Slide mode. Yes.
It stopped working.
Looking good to me.
Looking good? Okay.
Yeah.
All right. Professor Su just presented the global picture of the problems we are having because of the climate change. And of course, the food security is going to be a big problem in the near future. So my work is somehow related to developing the technology that we would need for programming plants. And in this regard, I would like to share some of my most recent work regarding the construction of synthetic gene circuits for programming gene expression in plants. So for decades, we have been engineering plants, and that is for improved growth and yield, resistance against abiotic stress, for example, heat or drought, and then biotic stress, for example, bacteria or viruses, and how it has been done. So it has been done through the use of the constitutive expression of the transgenes. But it comes with a cost, and the cost is the metabolic burden on the plant, silencing, and the deleterious effects on the plant health. So if we take a look to the tools that are currently available for engineering plants, so those are strong constitutive promoters, for example, 35S promoters that has been widely used in plants. Cell type specific promoters are inducible systems, for example, dexamethasone or the estradiol inducible promoters. However, all these different tools that we have currently available have certain limitations. For example, the strong 35S promoter is non-specific and is commonly silenced. The cell type specific promoters, they mostly can't respond to additional conditional inputs and are constrained by their natural range. And then the inducible systems, for example, dexamethasone, they are mostly ubiquitous and generally can't respond to multiple variable inputs. So we need new tools for more sophisticated control of gene expression. Why we need these ones? Because we need these ones to engineer smart plants that are capable of responding to multiple input signals such that when and where a response is required, so we can produce it. And how it is possible? It is possible with the help of synthetic gene circuits. So those who are not working in the area of synthetic biology, they might be wondering what are these synthetic gene circuits? So synthetic gene circuits are similar to the electronic circuits, which are composed of wires and transistors, but instead of those, these are composed of interacting biological parts such as DNA, RNA, and proteins. So here is a simple example of a promoter driving the expression of a reporter gene. So ideally, this reporter gene will be switched on. However, in the presence of an input such as a repressor, this reporter gene will be switched off. So this switching off of the promoter, of the constitutor promoter by a repressor is similar to an electronic NOT gate. So when we have the input 1, the output is 0, right? So now imagine a scenario where we have two different repressors, repressor A and repressor B, such that in the absence of these repressors, the promoter will drive the expression of the reporter gene. However, in the presence of repressor A, B, or both together, this promoter will be occupied and the gene will not be expressed. So this is similar to an electronic NOR gate, which requires two different input signals, input A, B, to perform its logical operation. NQ, which represents the output, will only be produced in the absence of input A and B. However, in the presence of input A, B, or both together, the output will not be produced. And we call it as a universal gate, the NOR gate, because it can be layered to make complex circuits. And this is something I will talk more about during the later part of this talk. So if we take a look, people have used different tools to create these synthetic gene circuits. For example, DNA binding domains, RNA regulators, recombinases, and the CRISPR-Cas9 system. So my talk will be mainly focused on the CRISPR-Cas9 system. So again, coming to that same concept of the NOR gate that I just explained, that taking a promoter that is driving the expression of a reporter gene, here the luciferase. So what we wanted to do, we wanted to target this promoter with CRISPR-Cas9 systems. I would like to mention here that this is not the usual Cas9 which cuts the DNA. This is a deactivated version which has lost its enzymatic activity. So what we wanted to do, we wanted to target this promoter with two different guide RNAs, which will serve as two different inputs like input A, input B, such that in the absence of any guide RNA, the Cas9 won't be able to bind the promoter. However, in the presence of input A, B, or both together, this promoter will be bound by Cas9, and as a result of the CRISPR interference with the RNA polymerase, there won't be any transcription. So when we don't have any transcription, there won't be any translation, and as a result, the gene will be repressed. So this is something we wanted to start with the NOR gate. To start developing this system, I took two different promoters from plants. One is the TCTP promoter, which is a moderate expression, constitutively expressed promoter, mostly used in Arabidopsis, which is the model plant. And then 35S promoter, which is the most commonly used promoter in plant sciences. So I took these two promoters and then introduced unique guide RNA binding sequences for Cas9 binding. Why we needed to introduce these ones, because the Cas9, it requires the PAM sequence NGG. However, most of the plant promoters, they are AT rich. So we wanted to engineer this one for the Cas9 binding to drive the expression of the reporter gene, in this case, luciferase. And then to test whether this promoter would still be would be able to drive the expression of the luciferase in the absence of the Cas9, we needed a fast platform for testing these engineered promoters and, of course, synthetic gene circuits, because working with plants, Arabidopsis, which is a model plant, but still to create stable transgenic lines, it takes months. So to develop this assay, I optimized the Arabidopsis protoplast system. So as I mentioned, Arabidopsis is a model plant that we are working with. And protoplasts, those who are not working with plants, these are the plant cells without the cell wall. So what we routinely do, we take the Arabidopsis leaves, like 4 to 5 week old, and then we remove the cell wall enzymatically and then introduce our constructs using polyethylene glycol. And as you can see, the architecture of this construct, the engineered promoter is driving the expression of the Rhinella luciferase. And then we have another constitutively expressed promoter, for example, NOS, that is driving the expression of the firefly luciferase. So this NOS promoter, which is driving the expression of the firefly luciferase, it is used as a normalizer for the transfection efficiency. So once the constructs are introduced into the plant cell, then we perform the dual luciferase assay, measuring the luminescence, which is produced as a result of the catalytic activity of these luciferases. So we are not measuring the transcript abundance here. We have compared the qPCR and the dual luciferase, which is the results are pretty much similar. So working on this principle, introducing different guide RNA binding sites upstream and downstream of the Tata box, we created two different libraries. One was based on the strong 35S promoter and one on the moderate expression TSTP promoter. And here you can see that we created a wide range of different strength promoters, and we were able to generate these promoters. They could still drive the expression of the repertory genes even in the absence of the Cas9. And all these different variants, they have unique guide RNA binding sequences. So to create the functional NOR gates, what did we do? We picked two different promoters from each library. For example, number 1 from the 35S library and number 3 from the TSTP library. And then we wanted to test their performance in the presence of the Cas9. And here you can see, in the absence of any guide RNA, the promoters are active, whether it is the TSTP or the 35S. And then in the presence of the input A, B together, in both cases, the promoter was quite repressed, and we were able to achieve up to 90% repression. So this was the first demonstration of the CRISPRi-based NOR gates in plant cells. It is just one slide, but the optimization and all the background work, it took us almost 3 years to generate this data. Now, going forward, those who are working with the CRISPR systems, they are aware that the guide RNAs are usually expressed from the PO3 promoters in the eukaryotic systems, which are usually the U6 promoters. However, what we wanted to do is to provide more sophisticated spatiotemporal control of gene expression. And for that, we needed to express our guide RNAs from cell type specific or inducible promoters, which in most of the cases are the RNA Pol2 promoters. So to expand the programmability of our NOR gates, what did we do? As a proof of concept, we took the 35S promoter to drive the expression of the guide RNA. And for that, we compared 3 different systems: the ribozymes, the tRNA, and the CSY4 system. The CSY4 is another CRISPR system, which requires like 20 base pair in the RNA transcript. To start with the ribozymes, so we took the guide RNA sequence, sandwiched it between the hammerhead ribozyme and the HTV ribozyme, and which are the self-catalyzing ribozymes, and putting this sequence under the 35S promoter. And then as a result, we could produce the mature guide RNA. So here you can see that we could produce a functional NOR gate, but the amount of repression we were getting was not as good as we saw with the U6 promoters. And then we moved to the tRNA system. Again, the concept was the same. The guide RNA sequence was sandwiched between the tRNA sequences and put under the 35S promoter to produce the mature guide RNA. And here you can see that we could create functional NOR gate. I would like to mention here that the promoter here we were targeting was the TSTP promoter, not the 35S. The amount of repression in the tRNA processed guide RNA system was really good. But unfortunately, here, I don't have the data here, but I can show it later, that we were getting pol 2 and pol 3 transcription together because the tRNA system has internal binding sequences for the RNA pol 3 promoter. So we switched then to the CSY4 system, which is another small CRISPR protein which requires 20 base pair in the RNA transcript. So here you can see these are the binding sequences for the CSY4. The CSY4 binds the cut of the 20 base pair at the 3' end remains there, but it doesn't affect the performance of the guide RNA. And as such, we could create a functional programmable NOR gate to express the guide RNA from the 35S promoters. We were quite happy with these results because we were able to express the guide RNA from the pol 2 promoter, which was the ultimate goal. Now, all this data that I have shown so far is coming from the plant cells, and ultimately, we wanted to see whether we can translate these results into stable transgenic lines. And that's why we wanted to test the functionality of these CRISPR-I circuits in stable plants. And for that, what did we do? We linked the guide RNA to express it from the dexamethasone indocible promoter, which is a chemical, and then the guide RNA B was expressed from the heat-inducible promoter. And we targeted the strong 35S promoter, which contained the binding sequences for these two different guide RNA to bind the Cas9 here, driving the expression of the luciferase, and this NOR gate was then transformed into Arabidopsis plants. And here I would like to mention that we were then measuring the transcript abundance using the qPCR. So here you can see in the absence of dexamethasone and the heat treatment, the promoter is active. And in the presence of the heat signal, we could achieve significant amount of repression. Unfortunately, the dex-inducible promoter didn't work. We tried different things, different concentrations, but it just didn't work. I'll get back to it later, that why it didn't work. So then we moved on to further characterize the performance of the CRISPR-I based circuit in stable plants. So we tested its performance in cotyledons. So you can see in the absence of the heat signal, the promoter is active. In the presence of the heat signal, we can see a significant amount of repression. Same was the case in the roots. And then we tested it in mature leaves. So here you can see in the absence of the heat signal, the promoter is active at time 0, but when we heat treated the plants and then tested the performance at 6 hours, so we were getting like significant amount of repression, and then after 24 hours, it started reverted back to the on state, and then after 48 hours, it was completely reverted to the on state, and then was stabilized at 72 hours. So to our understanding, this was the first demonstration of a CRISPR-I based reversible circuit in plants. Now, getting back to this dexamethasone promoter, that why it didn't work. So what did we do? Instead of driving the expression of the guide RNAs with these two different parameters, we switched them with the luciferases and wanted to compare the strength of these two different parameters. And again, we tested this in the Arabidopsis protoplast system, as I explained earlier. And here you can see in the absence of DEX in heat, there was no luciferase activity. However, while comparing the strength of these ones, so the heat inducible parameter was 5 times higher as compared to the DEX inducible parameter. This was suggesting that the quantity of the guide RNA produced from the DEX inducible parameter was insufficient for repression. And again, we were targeting the strong 35S parameter here. To further dig into these details, what did we do? We took different parameters of varying strength from the library that we had generated to drive the expression of the guide RNA. And then we targeted either the TCTP parameter or the 35S. And it doesn't matter whether it is like the moderately expressed parameter or the the strong promoter. In both cases, when the strength of the input promoter driving the expression of the guide RNA is reduced from 35S to a TCTP promoter, so we can clearly see that the amount of repression is dropping from 90 to 65% repression. So they suggested that the amount of guide RNA is a key design consideration for achieving effective CRISPR-I in plants. Then the next thing we wanted to see whether, because in the beginning of my talk, I mentioned that the NOR gate, we call it as a universal gate, so that we can then layer it in different arrangements to create complex circuits. This talk was about the NOR gate, where we were targeting the two different guide RNAs to a promoter that was driving the expression of the luciferase. So we wanted to create a programmable OR gate. So for that, we replaced this luciferase with another guide RNA. And this became our layer 1, and then targeted this guide RNA to another promoter, which had the binding sites for this guide RNA. So what we were trying to do that hopefully in the absence of input A and B here, this promoter would be able to drive the expression of the guide RNA C, and then that can bind to the layer 2 promoter to repress the expression of the luciferase. However, in the presence of input A, B, or both together, the expression of the guide RNA C from layer 1 promoter would be stopped. And as a result, the layer 2 will not receive the input signal and the luciferase will be expressed. So to test this in plant cells, we saw the expected results in the absence of any guide RNA promoters. The layer 1 promoter was able to drive the expression of the guide RNA C that was targeting the layer 2, and as such, the luciferase was repressed. However, in the presence of input A, B, or both together, guide RNA C was not expressed, and the layer 2 promoter was still able to drive the expression of the luciferases. And then we further arrange these NOR gates into different arrangements to create N imply B, B imply A, and AND gate. And it performed expectedly in the plant cells. So all this work was done in the model plant Arabidopsis. The next thing we wanted to see whether we could just simply import our constructs into different plant species. For example, moss, where we tested the performance of a full NOR gate. And these are the results from the protoplast, like the plant cells after 24 hours, without modifying any of these constructs. So the results were still promising. So we could see a significant amount of repression in moss. Similar was the case where we just tested the input A in wheat and also in canola. So these are the results from 24 hours protoplast, which were quite promising because we did not have to do any modifications. so which demonstrates the cross-species utility of this system. So in summary, we have developed CRISPRi-based circuits. They are quite modular, programmable, reversible, sensitive to input signal strength, and functional in various plant species. And we published this work last year in May in Nature Biotechnology. And they are being here. We got funding for this ARC Center of Excellence in Plants for Space. So we are using these CRISPR-based circuits for programming plants for space, mostly working on the small plants. So that hopefully one day we could go to Mars and we would need food there. And also not only Mars, as Professor Su mentioned earlier, on Earth there is also a huge demand of food. So hopefully these circuits could be of help to all these different requirements regarding the food security. So looking forward, the future development would be to make these circuits more robust and to do some modeling to make them more predictable, tunable, and interface them with the host regulatory pathways. So we can also introduce some more custom inducers and could also import into different plant species. So ideally, the large-scale synthesis and screening and the quantification and the modeling would be the new avenues to look forward into it. So not only us, Sarah Jesu from Arham Institute, she has done some beautiful work using the recombinases. Also, James from our lab has done some beautiful work using the recombinases on the synthetic circuits. And of course, Jean Brophy, she has done some amazing work on synthetic gene circuits for programming plants. And I would like to conclude my talk with it, and special thanks to all who had contributed to it, all the people and the funding organizations. Yep.
Fantastic talk. So why not? Let's go to the Mars to do agriculture or new habitat over there. Actually, we also published one paper potentially sending microbes to the Mars or Moon. Why
I think
not?
that would be a good
We
area to
can,
look into.
yeah, we
Like
can
some,
work together. Absolutely. Because I mean that's one of my dream as well. Not because my last name is Moon, but I want to go to the moon. Absolutely beautiful talk. Thank you. Let me ask Dr. Lee whether she has any question or comment. Uh, if not, I will start the question first. So you beautifully, demonstrated very important first step to engineer plants. And then that is beautifully, I believe I saw that paper published in Nature Biotech. But that's, I believe, still very first step to
yep.
get there. There means, you know, very routine engineering of plants. Even though, I mean, I would say plants engineering done many, 100 years, using simple breeding, etc. But still, our engineering, I'm not sure we can even say engineering. So to get there, I mean, what would be uh, remaining challenges, as you also already, imply or mention a little bit, but can you kind of elaborate potential challenges or opportunity to get there, like making plants like E. coli cell, we can do things very easily in the future.
That's a very good question, and some of the things we are currently looking into to get there. Of course, it won't be as fast as the microbial systems.
Right, that's problem.
Yeah, that's a problem. But the thing is, one of the major challenge that we are facing, because whatever you do in the best we could do, like to get like the faster results would be using the plant cells. but not all the time these results can be translated into stable plants. So whatever you see, because when you transfect protoplast, you cannot control the amount of the plasmid DNA going into the cells. And these plasmids are not chromatinized. And of course, plants are multicellular. So there are the different cell types, the environment is there. So those are the big challenges. So looking forward and going, and then the other challenges, because a lot of work has been done in the model plant Arabidopsis, the other crops, wheat, canola, all these different ones, people have just started looking into them.
Mm-hmm.
So the cell type specific promoters and those regulatory regions, so that would be something to get started with it, characterizing all these promoters. But again, it is going to take a lot of time. One of the avenue would be to produce lots and lots of data and then do some machine learning. But again, the problem and the challenge would be like today, one of our review was just published. So some of the aspects that we had talked about, the challenges in there. And so, yeah, so those are like the major challenges. But hopefully, plant synthetic biology people have started looking into it more and more. So maybe one day we would be able to address these challenges. Of course, not as fast as like the microbial system.
Mm-hmm.
Lots and lots of data and those modeling that would be quite helpful to some extent.
Sure. So absolutely. So that's very interesting. To me. Uh, because also I heard many time from plant scientists or engineers. Uh, Interestingly, Arabidopsis is kind of used because of convenience for many years, but particularly that plants is not that much useful. Because like wheat or rice. This is more useful, but not that much research done in terms of engineering before. That is actually too interesting to me. Probably because of the tool is not available
Yep.
for those, one. So hopefully, I mean, in the future, we have the more tool in that way, we, routinely engineer. That might be another reason I not that brave enough yet to enter plant science because I'm very impatient in terms of waiting for result because I'm start my career with the bacteria E. coli and then now I'm moving into the non-model organism because I realize E. coli is not that much important now because we already done a lot in the past. So I'm moving into that one and potentially moving into the plants in the future. Um, Actually, do you know why, people just use Arabidopsis because of other than the tool available or, it's easier to deal with compared to other, other plants?
That's a very good question. And the thing is, like, I mean, um, when it comes to publishing, of course, like for a good academic career, publications matter.
Mm-hmm.
So, because working with Arabidopsis, the life cycle is short compared to other
Mm,
plants. One of the major reasons that people want to work
got
with it.
it. That
So
makes
that's,
sense.
yeah. And then when you are starting to work with plants, when you send your paper for publication, so let's say you have, so two of the main methods that people use is either working on the plant cells, the protoplasting, or the agro infiltration, like using the benthamine, so where you take the agrobacterium culture and inject into the tobacco leaves and then study it. So that cannot be just high throughput. That is like a bit more laborious because first you have to do cloning in E. coli and then taking it into agrobacterium and then syringe infiltrations. But if you have done like any tool development or any studies, so reviewers would still ask you whether these results can be translated into real
Uh-huh.
plans. So if you are working on wheat, I'm not sure how long that is going to take.
That's true. That's true.
That's why people are just sticking to Arabidopsis
Uh-huh.
because though it is not the life cycle is short as like the microbial systems, but compared to the wheat,
Uh-huh.
where the engineering and then the genome is not as simple, it's quite complex to
Got it.
overcome those hurdles. So people are just sticking to Arabidopsis, and now particularly people are looking into these simple plants, for example, moss,
Uh-huh.
which just show that result from, and then also Markentia. So Jim Hasselhoff has done a lot of work at Cambridge on those model plants. So they are like even shorter and simpler,
I
like
see.
Arabidopsis.
That completely makes sense now because, when reviewer ask revision experiment, if that takes 6 months or 1 year, I mean, it's not gonna be also without guarantee whether that experiment working or not. It's kind of not easy. There is no easy way to handle those kind of comment. So that means, I mean, the academic culture should be changed to encourage more young people to enter that field. Otherwise, it's almost impossible to make this field is a more active and so on. But I now optimistic because a lot of young people start their career, including some people I already know. I mean, mostly uh in US because those people I know more. But I know multiple speaker actually start the plant scientists in these cities. Jennifer, Jen Brophy, started many years ago in at Stanford, goes there, et al. I mean, those people is doing fantastic, and then including you as well. So I'm pretty excited to see you guys kind of doing fantastic job.
Thank you.
Let me double check one more time. Dr. Lee, if you are still there, I wanted to give you a opportunity to ask question or comment.
No, I think you asked all the questions that I wanted to ask, so I'm good. Thank
Sure.
you so much. Great talk.
Thank you very much.
Fantastic. So in that case, I mean, let me double check whether we have question from audience. Okay, I don't see any. Uh, we close because it's almost on time. Let me close and then we probably have potential, chat afterward. Okay. Thank you all for joining and staying today. We'll meet again on March 6th, Thursday, the same time, the same Zoom link. We'll have Dr. Tammy Collins, a program officer at Borough Welcome Fund, and Elena Mustiata at Rice University. As usual, the follow informal chat will occur without recording. Please stay here if you're interested in chatting with us. I'll promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thank you again. I've stopped recording. Just 1 second.